Shall we start? Ah, uh, Today I am Dr. Vittal from head of the Department of Microbiology, Himayatnagar branch, and now today we have a live discussion as well as a webinar on human metapneumovirus infections presented by one of my colleagues, Dr. Rizwan Ansari. He is working as a part-time microbiologist at Himayatnagar Vijaya Diagnostic Center. And Dr. Rizwan Ansari is working presently as Assistant Professor of Microbiology, Clinical Microbiology at Apollo Institute of Medical Sciences and Research. He has done his undergraduation, that is MBBS, from Deccan College of Medical Sciences, and his post-graduation, he has done it from Kakatiya Medical College, Warangal. Presently, he is working as assistant professor, and he is also associated with Telangana Antimicrobial Surveillance Network in collaboration with National Disease Control Center. And today's topic of human today's topic of human metapneumovirus infection is presented by Dr. Ansari. Now we'll start the session, and I'll hand it over to Dr. Ansari for the presentation.
Good afternoon, everyone. Am I audible?
Yes, Dr. Ansari, you are audible.
Yes. So, once again, good afternoon, everyone. And I'll start with extending warm thanks to entire VTC team, especially our lab director, for giving me this opportunity to speak on this platform on the topic human metapneumoviruses infection. So, moving ahead, human metapneumovirus is not a new virus. It has been there since humanity, but it was discovered in 2001 by a group of investigators in Netherlands, and they discovered that this virus is associated with the respiratory disease in children, and then they named it as human metapneumovirus. Okay, but clinical data suggests that this human metapneumovirus has been there in the worldwide in the last 60 years. It has been causing infections worldwide in distribution. Moving ahead with the virology, I won't bore you, but small note how to know this virus. So it is belonging to a member of family Pneumoviridae, and it is a large enveloped virus with having a negative sense RNA virus, and it was previously under a family Paramyxoviridae as a subfamily there in a, now it has been reclassified in 2016 as a separate family with two other genera. So one genera includes metapneumovirus, which includes the human metapneumovirus itself, and the other genera is the orthopneumovirus, which includes the respiratory syncytial virus. Next, it's an as we said, it's an enveloped virus and has a non-segmented negative sense RNA as a genome, and it is closely phylogenetically related to avian metapneumovirus. So avian metapneumovirus, it is related. So, but the scientists thought that it could have been originated from the birds, but in fact, it is an animal virus, and it has been seen that it causes infection in animals, including humans. Okay, so yes, so next, phylogenetic analysis revealed that this virus has two subgroups, subgroup A and subgroup B, and within each groups there are other two clades. So it one A1 and A2, then B1 and B2. And these four variants, they cause concurrent infections, and there is no study established to say that one variant is more virulent than the other virulent. And annual circulation of we have seen that annual circulation of all the four variants have is there, with one one which is more predominant, and every year it changes. Say if A1 was predominant in a particular year, then next year A2 is predominant, then the B1 is in the next year, like that. So also the human metapneumovirus inoculation done in monkeys has led to significant viral replication restricted to the respiratory tract, and subsequent development of the upper respiratory tract symptoms. So this lead to know that the viral pathology is limited to the respiratory tract. Next, coming to the viral pathology, as we have discussed, it's a respiratory tract invading virus. So the point of entry in the respiratory tract is integrin alpha V beta 1 cell surface receptor. So it is a cell surface receptor present on the epithelial cells, and this receptor is having various functions like adhesion, signaling, interaction with the extracellular matrix and all. But the virus HMPV uses this receptor as a point of entry to gain entry into the epithelial cells, and then leads to infection, which leads to hyperproduction of mucus, hyperplasia of the respiratory epithelium, and resulting in the airway obstruction and hyperresponsiveness of the airway with methacholine challenge. So these changes produced by the virus are the main reason for the pathology or the pathogenesis or the clinical manifestation. Now, how it is transmitted? So transmission occurs by direct or close contact with the contaminated secretions or the respiratory secretions. Physical separation at a distance of 6 feet therefore minimizes the aerosol transmission, which we have seen during the fight in the COVID-19, right? And one study of school age children has revealed that HMPV estimates for about 12% transmission rate to the household members. So this is usually seen in school going children, and when they are infected and when they come back home, they are the index case for their household contacts. Nosocomial infections have also been reported in hospitalized children and adults. Coming to the incubation period, the incubation period, it is approximated to be around 5 to 9 days. Transmission from family members appeared to be very efficient when the index case has become ill. So the time interval between the onset of symptoms in the index case and the contact is around 5 days. That means once the virus has infected the index case, once the virus has infected the index case, and uh, when the symptom starts in the index case, from there to the symptoms in the contacts, it takes around 5 days. So this establishes the incubation period of 5 days. Coming to the epidemiology, virus is ubiquitous. Ubiquitous means it is present everywhere. So as it is ubiquitous, there is worldwide distribution. Global burden of this disease is very large, but um, and this virus has been associated with infections or with us at least 6 decades, and it causes upper and respiratory tract infections in all age groups, but symptomatic disease is limited to young children and older adults. Means those extremes of the populations which are having less immune defense. Coming to worldwide, the models have estimated that approximately 14 million human metapneumovirus associated LRTIs are seen under the in children under age of 5 years. This data was around on 2000 in 2018. Next, HMPV shows seasonal variations. So in temperate zones like America and UK, so late winter and early spring are the more predominant seasons where this infection prevails, whereas in Asian regions like our country, China, Hong Kong, so late spring and summer are the main seasons. Coming to the clinical manifestations, we'll divide the clinical manifestations in children and then adults. So first we'll go with the children. So the common symptoms include cough, fever, rhinitis, sore throat, body ache, and headache. Whereas the if so these symptoms, they are usually of upper respiratory tract, and when the disease is severe, then it resorts to shortness of breath, wheezing, chest pain, severe fatigue, dehydration, and dizziness. So this on the right hand side, the symptoms, they are mostly due to the lower respiratory tract infections. Now HMPV is usually associated with children, and it's a mild and self-limited respiratory infection in children and adults both. But the typical course of a mild disease, if you're talking about mild disease, is an asymptomatic period of several days after the infection has been acquired, and then a week of upper respiratory tract symptoms, which we have seen, like fever, cough, and then gradual resolution on its own or with some supportive therapy. Then peak viral shedding is seen at the end of the first week of after inoculation or after acquiring the infection. Peak symptoms will start after a day or two of the peak shedding. So peak shedding is when the symptoms are at peak. A day before or two, the virus, the index case or the case starts shedding the virus so that it contacts the other individuals. Coming to the course of severe disease, so it is mainly the lower respiratory tract infection, a brief asymptomatic period after inoculations. We have seen that in upper respiratory tract infection or mild cases, the asymptomatic period is large, whereas here, in contrast, the asymptomatic period will be short, brief, and then 1 to 2 days late, 2 days upper respiratory tract symptoms will be there, followed by progression into wheeze and other signs of lower respiratory tract infections. Now, this disease can show more prolonged course in the individuals with predominant predisposition with the reactive airway diseases. So, like prolonged airway dysfunction will be there, recurrent mild wheezing will be present, or cough for days or even several weeks are seen in individuals which are having reactive airway disease like COPD and asthma. Now, what are the patients where, what are the conditions where hospitalization will be required? So patients will require hospitalization if the disease is progressing into, LRTI is progressing into bronchiolitis, or the asthma, if the individual is already an asthmatic, there is an exacerbation of the asthma, or there is severe pneumonia, or there is development of the acute respiratory dysfunction syndrome. Sometimes only 2 or 3 rare cases have been seen of uh, encephalitis and sepsis syndrome. And in case of encephalitis, the RT-PCR done on the CSF fluid has revealed the presence of the HMPV RNA present in the CSF from the encephalitis case. Now, coming to the adults, so manifestations are somewhat similar, but fever is seen only in 4% of the cases, and majority of the symptoms are like dyspnea and wheeze, which is more common in adult patient, while hoarseness is seen in young adults. Other constitutional symptoms like nasal congestion, rhinorrhea, and wheeze are also present. Now, there has been discussion whether there is a possible role of COPD or exacerbation or asthma exacerbation due to HMPV. So, The exacerbation of COPD due to the HMPV has a conflicting results. Few studies have shown that it causes causes the exacerbation of the COPD, whereas few studies have shown that there is no such evidences. Then the asthma exacerbation has been proved with a positive association by HMPV and various studies. Now, coming to the recurrences, what are the chances of recurrence? So, recurrence is very common and appears to be mild in majority of the immunocompetent patients. But coming to the immunocompromised patients, they are at a risk of getting a severe disease when they are having a recurrent infection. Antigenic drift likely occurs in some extent with the HMPV, but is not necessary for reinfection. So, similar variant or similar strain can cause reinfection. There is no need for antigenic drift that has been established. Coming to the clinical manifestations of the immunocompromised host, so frequency of acquiring the infection appears to be same as that of the immunocompetent individuals, whereas the disease progression or the course of disease is more severe and the course is more prolonged in case of the immunocompromised individuals. And this is attributed to the poor clearance of the virus after infection. Coming to diagnosis, so first thing is, why, as it is a self-limiting condition, why do we need to diagnose? So need first point will be, we have to achieve a specific diagnosis of HMPV and in severe respiratory tract illnesses, so to avoid unnecessary use of antibiotics, as we have seen that the respiratory symptoms are similar to that of the other respiratory viruses and few of the bacteria. So the clinicians need to know that whether it is a bacterial pathogen that is causing the infection or a viral pathogen that is causing the infection. So if it is a virus, then the use of antibiotics will not be required or it is unnecessary. Second thing is, if we know that this virus has been HMPV has infected the patient, we can take proper precautions to isolate the patient, as it is a contagious disease. Coming to these specimens, respiratory secretions of all kinds, sputum and the bowel or bronchial brush and swabs of the respiratory tract, oropharyngeal tracts are the specific or the selective specimens. Serum routine testing is not done, but used in case of other research purposes. Now, what are the testing methods? So, first is the RT-PCR. Thanks to our friend who has discovered the PCR, Kerry Bimulis. So, most sensitive method, both conventional and real-time formats are available. And then we have a biofire film array, which has in this the respiratory panel, the HMPV has been included and it is FDA approved. Then we have the direct fluorescent antibody detection of the detection of the viral antigens, and then viral cultures. So viral cultures, only 50% of the RT-PCR positive cases yield cultivable viruses. So it is not very much sensitive. Coming to the RT-PCR, so the gene which is usually targeted is the nucleocapsid gene or the fusion gene. So fusion gene is the responsible for the fusion of the virus to the epithelial cell receptor. Serology, HMPV specific antibodies can be detected using ELISA format, but it is reserved only for epidemiological and research purpose. And to establish a serologic diagnosis, two things is required. Either should be a zero conversion or at least four-fold increase in the titers when serial samples are being tested. Only then we can establish a serologic diagnosis. Imaging is non-specific. Most of the common things will be showing like perihilar opacities, hyperinflation, atelectasis, and occasional consolidation, but there won't be any pleural effusion or pneumothorax. Similarly, peripheral blood counts, the early stages of infections, show a reduced number of peripheral blood lymphocytes and elevated monocyte ratio. But all these findings are not specific and cannot distinguish bacterial infections or viral infections or HMPV from other viral respiratory viral infections. Coming to this supportive treatment is mostly supportive and it varies with the clinical manifestations. So depending upon the severity of the disease, treatment will vary. Antiviral agents like ribavirin, which is found to be active in vitro, but there is no clinical data suggesting that efficacy of ribavirin or any other antivirals for HMPV infection. So brief note about ribavirin: it is a guanosine analog, so it halts the RNA synthesis, and also viral mRNA capping is seen. It is a drug of choice in case of RSV and hepatitis C virus. So therefore, no antiviral therapy is recommended for patients with HMPV infection. Next, antibiotics. So as we said, it's a viral infection. If we have the established diagnosis of viral infection or HMPV, then antibiotics are not required. So secondary bacterial lung infections or bacteremia patients um, also have shown. So there is no studies that are shown established secondary bacterial or lung infection associated with the HMPV. So use of antibiotics in hospitalized individuals or ICU individuals is also not recommended if the diagnosis is established as a HMPV. Coming to the prevention, infection control measures are warranted if the case is suspected or confirmed case of HMPV. So, what are the infection control measures we would put forth? They are like what we do for RSVs or any other acute respiratory viral infections: hand washing, use of gloves, and appropriate infection control measures like surgical mask, eye protection if there is chances of splashing of the healthcare workers face with the infectious respiratory secretions. Then standard and contact precautions include also the isolation of the patient in a private room, if possible, or if there is an outbreak, then cohorting the patient with HMPV infected patients. And should one note is that HMPV infection can cause co-infection with RSV infection. So, and there are studies showing that co-infection of RSV and HMPV has led to a bad outcome or guarded prognosis. So, there is a thing to not combine the HMPV infection and patients with HMPV infections and RSV infection in a single room. Then immunocompromised patients, as they are at higher risk, they should avoid contact with the children in the acute HMPV infection or in early convalescence period. And last thing is vaccine development. So the first thing, there is no vaccine right now available, but there are live and subunit experimental vaccine candidates that are still in preclinical development stage. One candidate is recombinant human metapneumovirus VF, human metapneumovirus F. uh, Candidate F stands for fusion glycoprotein. So this fusion glycoprotein uh, is the protein that is responsible for fusion or entry of the virus inside the epithelial cells. So if we can target this particular fusion glycoprotein, then we can prevent the virus entry into the epithelial cells. So with this, I would like to end my talk on HMPV.
Okay, thank you, Dr. Ansari. If you have any doubts, the session is open for clarifications. Anybody who wants to ask? Anybody? Any doubts? Okay. Nobody has any doubt. So, can we conclude the session, Mr. Ranjit? Thank you, Dr. Rizwan Ansari. Thank
Thank you,
you.
sir.
Thank you, sir.